somehow the recordings. There we go. Shefali, so I'm sorry, what high school is it?
Del Norte High School.
Which one, sorry?
Del Norte High School.
Del Norte, okay. And then tell me a little about your, what level you're at, Flora, and where? So it's also Del Norte?
So all of us are on the Del Norte High School iGEM team, and we reached out because Anushi and I did the internship, like the smoke-free project with American Lung Association. And so we asked Pierre, and Pierre redirected us to you.
Got it. Okay. So smoke-free project from the ALA, and then Kara, yeah, I'm trying to remember, had referred. And then Anush, there's somebody else that
Anushi also did the project with me, but she said, I don't think she was able to make it today.
Yeah, no problem. Okay, great. And have you done a few people like me interviews, or am I the one you're catching? I don't know.
I think we're starting off with you.
Okay, you can get more as you go. Okay. All right, go ahead, sorry.
Okay, no, you're good. Okay, so I had a couple of questions on your own research to start off with. So one of my questions was, were there any anomalies in your study where like these sugar molecules weren't as accurate as you thought that they would be?
So where they were not as accurate?
Yeah.
So you have to be a little more specific. Accurate in terms of what? Like what?
Identifying the cancer in the system.
Identifying what? Sorry?
The cancer in this is like the lung cancer.
For in association with lung cancer. So there's a lot of expected and unexpected things that come up. Maybe it's better if I just back off a little and tell you how we study sugars in cancer, because it's not always in my research. And just to give you it in just a 1 or 2 minute overview, what we are studying is we have a lot of interest in complex carbohydrates on the cell surface. So that's different from protein, different from lipid. And there's a whole world of glycobiology where carbohydrates play very important roles in making contact between receptors, in internalizing molecules on the surface and turning over the cell surface. And in cancer, directly on the cancer cell, there's a number of functions that helps the cancer cell spread, to repel from among cancer cells during metastasis. Just to give examples. And then there's and many other roles that help a cancer cell do its cancer spreading thing. There's a bunch of other roles, and that's where we spend actually more time in my lab, of how sugar molecules are involved in regulating immunologic processes that are involved in controlling cancer by your own immune system, and also involved in my own research in years past in controlling the other host response to cancer, which is vasculature. So investment with blood vessels and investment with lymphatic vessels, which are the ones that drain to lymph nodes. Those are indirect. We'll just say they're more the interest in how sugars are in host elements, your own body's normal host elements that have to do with blood vessels or immune cells or lymph nodes and all that stuff that is around the cancer, and trying to do something to control or limit the cancer. So there's kind of two sides we study, and we spend a lot more time on the host in my lab than on the cancer cell itself. But I'm happy to share with you some interesting things we study in the cancer cell and what I've studied in the past on the cancer cell, because there's still quite a bit there. Does that make sense?
Yeah, that does. Thank you.
Yeah.
I had another question. So how is your research like implemented in curing cancer today? Like, how is it used to help?
So good question. So we're at very early steps in our work. We're basic scientists. I'm a basic scientist. My most direct link to reaching patients one day is really understanding better how we can help the cells in your body in a tumor or near a tumor that present parts of the tumor to T cells, to your immune cells, do a better job at presenting those, so we attract more anti-tumor cells into the tumor to help kill the tumor. There's been some, if we can make that work better, and we can take something that helps the sugars do something in a way that promotes that that presentation of foreign antigens from the cancer cell to the T cells, we've done a good thing in the lab, because those kind of things can, if you learn the basis of that, you can translate that into therapies one day that someone can take to help their immune cells counter the cancer better. That's basically the long and short of the most direct thing. I don't think I answered your first question very well, which was, I gave you an overview, but your question was, do we see kind of things we don't expect or irregularities in the sugars? And I just wanted to finish by saying, yes, we often run into what we don't expect, and we don't have a good feel for, like in chemistry, you deal with stoichiometry with quantities. We're dealing with sugars on a cell surface. So unless we take those cells out and we have, I can say, yes, we do have specific ways that we can genetically lower the sugars and see what that does to cell behavior on a dish, then we have a good control. But once we go into 3D in the body, because we work with mice and we work with models where we have to understand how the tumor biology is being gauged or changed by changing the sugars, we don't have as good of a feel for a kind of stoichiometry or a mass effect. Like, do we have to affect 30% of the cell surface or 20 or 80% of the cell surface to have the effect we want? Does that make sense?
Yeah, it does. Thank you. Also,
Yeah.
Hannah, did you want to?
Yeah, I just wanted to interject a little bit. So kind of before all of this came to be, is there any important prior work we should know about, like in the field, or is your work considered novel?
Yes, you probably the most direct work to help you understand where we're coming from. It's a great question, because I want to give you a basis that we are by no means shaking the world the way my lab isn't yet. The way that cancer immunology people have shook it in the last maybe 5 years. So there have been many decades of trying to help the host respond to cancer better, but it failed. There was never really good vaccines for cancer. There weren't good cellular therapies, but there was a Nobel Prize a few years ago, as you guys may or may not know, that discovered a way to block what are called immune checkpoint pathways. And what they are natural pathways where T cells can sort of engage with foreign things, and the foreign thing being a bug or a cancer cell or anything it is, and make that T cell attack better. So immune checkpoint pathways are, the best way to explain it is they're sort of homeostatic mechanisms to help T cells stay quiet and not get over ramped up when they discover something foreign. Like, for example, a molecule being presented on one of these antigen presenting cells in the tumor. Those are the ones we study. We're very interested in helping that work better. And what the Nobel Prize discoveries have done is, and they're in our clinic now, we have it as therapy, what are called immune checkpoint therapies or immune checkpoint blocking therapy, is they're antibodies that block this kind of a turning off switch between the T cell and the cancer cell that engages with specific receptors that are immune checkpoint receptors between the tumor cell and the T cell, or the antigen presenting cell and the T cell. So it's a little complicated, but said briefly, it's a mechanism that your body has to basically always shut off to some extent or put a brake on T cells when they engage with these molecules on cells where they're normally going to turn on to something foreign. And what cancer people have done in these Nobel Prize therapies is developed antibodies that block that immune checkpoint so that it helps the T cell get a little more excited and a little more unregulated in its growth, which is a good thing, when it sees the foreign tumor cell expressing that antigen, or the dendritic cell, which is the antigen presenting cell, see that foreign antigen being presented. They get more growth, and you grow the killer T cells in bigger numbers in the tumor that attack the tumor. So that's very novel. And that's the basis of immune therapy that has made big strides in the last several years in lung cancer and in melanoma. Those are probably the two biggest types of cancers that express a lot of foreign molecules. and we never knew a way to turn the body onto them. And basically those discoveries have revolutionized cancer care for advanced lung cancer patients that have a lot of metastasis. You can't do surgery. You can't, it's very hard to cure them with radiotherapy or chemotherapy. And chemoradiation is often what has been used historically up until these discoveries. Now we can use pure immune checkpoint therapy in many patients that qualify for it. And amazingly, hold a cancer in check or even cure a cancer in some cases with no use of chemo or radiotherapy. So all you're doing is helping the host attack the tumor, and you're not touching the tumor cells directly. So that's the background I would give you. And all we're doing is trying to help that work better as one of the things in my lab.
That's fascinating. Thank you for sharing. So this is just an assumption, so feel free to correct me if I'm wrong, but is this immune checkpoint therapy more specific than, let's say, other types of traditional therapies that have been used for lung cancer?
So basically, I will say that is one of the fascinations of my laboratory is we see immune checkpoint therapy as relatively non-specific. And what do I mean by that? The antibodies that they've generated are quite specifically against molecules that have names like PD-1 or PD-L1. Those are two, it's a ligand and a receptor that happen between the T cell and the tumor cell, or the T cell and the dendritic cell, which is your immune presenting cell. That's the handshake that shuts off the T cell. So they make antibodies to that, put them into the patient, and that blocks the brakes. So it accelerates, in other words, the attack of the T cell to, or the growth of the T cell to this foreign presentation of antigen. But those are kind of non-specific in the sense that they're not geared to see the antigen itself. They're kind of secondary pathways that are around what I call an epicenter. And the epicenter that we're interested in, and a lot of people in the world in research are, in cancer research, is, when I say epicenter, I'm thinking like an earthquake. Where's the action happening? Where's the center of like all the action that you want to make work, a bigger earthquake to kill cancer? And the center handshake that happens is between something called the T cell receptor and something called antigen with MHC1. So MHC1 is just a molecule that is a part of your repertoire on your cells to present foreign material on that MHC molecule. And the foreign antigen that gets presented with MHC sees the T cell receptor, and at that handshake is where the big stimulation happens to the T cell to start dividing and recognizing something as foreign, as long as it's had a chance to learn about it before. Often in a lymph node or in a tumor, they're naive, the T cells, but they have to learn about this through antigen presentation. And once they start learning something's foreign, that handshake is the center. All those other things that are called immune checkpoint pathways, they're kind of outside it, and they help to either stimulate the handshake or even turn it off. So there's brakes and there's accelerators. And they're not as specific as the actual foreign antigen piece, which is unlike your body and looks like a bug, or looks like it might be overexpressed by the tumor and just put on that MHC as a normal molecule, or it's foreign, like a bacteria or a virus. And you do this with viruses that attack you, COVID, flu, they get put onto MHC1, and the CD8 T cell sees that on their receptor. And that's a way for that T cell to get angry at a virus or get angry at cancer. So to answer your question, which is a really good one, yes, actually, I'm a believer, and you can say in some ways that immune checkpoint therapies are quite specific because they're targeting that PD-1 or that PD-L1 co-receptor, but it's really not the center pathway that is really the key specificity in attacking something foreign in cancer. So if we can help that epicenter work better, and we know that carbohydrates can do this, if we change or undersulfate some of the sugar chains near that epicenter, we actually improve the handshake. And if we can make that work, then you can keep up with any foreign antigen as the tumor evolves over time and starts expressing new things. You don't have to, you can just help that dendritic or that antigen presenting cell work better at any time in the future with the T cells as the cancer evolves, if that makes sense.
Yes, it does. Thank you for answering that.
Sure. Yeah.
All right. One last question from me. I'm very much taking over Shefali's show right now. But I wanted to hear a little bit about what your lab works on when it comes to vasculature that you mentioned earlier. So we talked
Yeah.
about the immunologic processes, and I'd love to get more into a second.
Yeah, the vasculature, it's I'm not as active at the moment, but we've published quite a bit on vasculature in the past, and glycans and carbohydrates. So glycans is a name for complex carbohydrates. And what we studied was cell division of what's called the endothelium. So it's the inner lining of blood vessels. And those when you have an injury or you have a tumor, anything that excites new remodeling of blood vessels near an area of either injury or cancer often makes molecules that are growth factors that drive those blood vessels to grow into the tumor. That's a process called angiogenesis. So it's generation of new angio, which means blood vessel. And there's also new molecules made by the tumor that drive lymphatics. That's called lymphangiogenesis. There's two kinds of endothelial cells. Those are the cells that line the inner lining of your blood vessels in your body, or the lymphatics, which are basically these networks that collect an ultrafiltrate of your plasma and bring it back to the circulation from the extremities and run through lymph nodes. So that's a way that the body has to run the lymph to these centers where foreign things can drain and the immune system can respond with T cell responses. So we've always had fascination with how those endothelial cells that make up that vasculature, those two kinds of vasculature, grow around tumors and how they grow in response to the tumors. And the way the sugars came into that are that, again, those glycans or complex carbohydrates on the surface of those endothelial cells are very important to help the handshake between the growth factor that's coming in to stimulate the growth of the endothelial cell and the receptor on the endothelial cell that drives division and remodeling of that blood vessel cell, that endothelial cell. So again, in that sense, carbohydrates, complex carbohydrates, very unique ones, molecules like heparan sulfate or chondroitin sulfate, they're complex carbohydrates that work as co-receptors to help those handshakes between the growth factor and its receptor that drives the vascular growth. So it was kind of fun. We could develop genetic methods to change those sugars, and we got reduced growth of vessels to tumors and smaller tumors. It's just a proof of concept. People have, as you can imagine, in the past have developed antibody therapies to block the growth factors and starve tumors of blood supply. And that's had moderate success, but it's not quite the same level of success as overall what chemoradiotherapy has done or overall what immunotherapies are doing now in lung cancer.
And you touched on using genetic modification to change how these complex carbohydrates behave. So when you talk about gene therapy, could you go a little bit more into specifics about how you see this being implemented and how you use it in lab?
Right. So we're really not doing real-time gene therapy. So we are not taking a vehicle that will bring a genetic change to cells in the body in real time as a tumor is evolving. What we're doing is we're using tools that control, there's systems that we have that allow for some technology, genetic technology that's pre-existing in a mouse. For example, we use a mouse that can cut the gene of interest or make a SNP in its importance genome in the exons that encode an important molecule that is involved in carbohydrate biology or biosynthesis. So for example, we have, for example, a way to control in a mouse in certain cells of the body, the molecular scissors that make a cut in the exons of an enzyme that sulfates the glycan chains or the sugar chains, the polymers of this sugar. And if we turn on that gene, which there's some technology to do, we can actually have the scissors that make that cut in specific cells of the body. We can do bone marrow cells, we can do blood vessel cells. We talked about blood vessels. So we can drive the genetic change in a mouse at very specific times to turn off, for example, the sulfating capacity of carbohydrates in that tissue. So in blood vessels only. So that's kind of nice because it's kind of select. That's not something that you can construct a human to have the same genetic technology and do that easily as gene therapy. We do it purely for learning, so that when we create that genetic change in the animal, let's say I want to undersulfate those dendritic cells, those immune presenting cells. So I do it with very specific molecular scissors that are driven by, that cut that gene only in cells where the promoter is produced that drives those scissors by a cell that's overexpressed in dendritic cells, or a molecule that's overexpressed in dendritic cells. So that pathway drives the scissors that cuts the gene only in the dendritic cells, and we get to watch how cancer biology in a mouse progresses with that mutation, and specifically in those cells. And what we learned from it is that when we undersulfate those sugar chains by specifically controlling it in those cells, which you could never do with a drug and just toss it into the system because it goes everywhere, it affects all cells, it's messier. By doing it genetically in select systems, we can, and we have technology for it, we then learn how tumor biology progresses. And what's fun is we can actually take cells out of that system where the mutations happened in the cells we want, and study them in the dish, or study how the T cells coming out of that animal would have responded to kill tumor cells, for example. If we take them out of the tumor, we can do it in the dish and see that when that mutation was there, the T cells kill better, for example. So there's a lot of in vivo and what we call ex vivo proof of concept, learning biology. And so therefore, what have we learned from that? Well, we've learned the importance of that enzyme that sulfates the sugar chains in controlling immune responses against the tumor. And when we learn that, your natural question would be, how do you get this to genetic technology to help a human with cancer? You know, how can we get it the next step and develop a therapy from it? And that's where we learn something basic in basic science, and the next steps are translating anything that can come as closely specific to that as we can. It's messy right now. There are gene therapies. There's called CRISPR technology. There's ways of getting, in vivo control of gene synthesis in specific cells, but it's tricky and not easy to do. There are things like antisense oligonucleotides. Those are in the clinics to control neurologic diseases, to control all kinds of disorders in molecular processing, people that have genetic disorders that overexpress something or underexpress something. Well, in cancer, if we want to change the expression of something in a specific cell type, we can use those, for example, antisense oligonucleotides. These are just short molecules that you can just put in the body, either through an injection, or you can imagine through inhalation, for example, to control molecules in asthma or even lung cancer. These are future therapies that potentially could work to get the environment changed, where you affect those same cells in the microenvironment you want, even if the side effects already affect some other cells in other, with the same genetic change. It's about winning the battle and getting those immune cells to work better. So a little messy in terms of trying to be stealthy with gene technology therapeutically, but in terms of learning, where we have the tools, we can be very stealthy. We can learn basic biology from it. And that's the goal. We're trying to figure out how the systems are working and how these glycans are controlling steps in immune biology. And as technology changes in the future, we can probably deliver the technology very stealthily to very specific cells with molecular targeting. But that's not there yet.
Someday. All right,
Someday.
I'll turn it over to Shefali to ask some questions.
Yeah, okay, so I had just one more question before we move to our project. So you said in your part about how there's more upcoming studies because you guys have to test on tumor DCs and et cetera, since that might have a direct correlation. So are there any updates on that so far yet?
Yeah, you mean new work that we're discovering on helping the dendritic cells work better, right? Yeah, we have the kind of newest stuff we've done is in the last couple of years in that specific area. We've published a couple of papers And we have new work that's coming from both cancer biology, where now we have models in cancer in mice that are spontaneous tumors. So we used to implant tumors where we can put a set of tumor cells under, you put the mouse to sleep, you put some tumor cells under the skin, you let it grow into a little tumor. We don't like survival studies. I'm not a believer in letting mice, I don't want mice to suffer. So we have very strict committees that allow us to do certain things on the mice, which is good. And there are survival studies where you let mice get cancer and could potentially die of cancer or live based on your therapy. I don't push mice to that limit. We are very strict about controlling an endpoint, which is time. For example, a little nodule growth where they're just eating and doing their thing, and you're just watching a little nodule of cancer under the skin grow from a few millimeters to a centimeter or something. And then we can euthanize the mice and do the studies. We actually understand what's happening to the biology. Well, now we have spontaneous models where the mice, we don't have to implant those. They actually have genetic technology where we can actually get spontaneous tumor growth to happen in the lungs. And we have very small tumors that can be generated in that way. And we're looking at the earliest stages of tumor growth. And so that same genetics that control the sugars on antigen presenting cells, they actually were fascinated with new findings on macrophages, which are some of the also antigen presenting cells, but they also kind of work in the lung and eat debris and work in that environment. And they put out certain cytokines or molecules that can affect early tumor growth. And we're actually learning that these mutations can change the amount of early nodule development. So at its earliest phases, even before the immune system's working. In terms of dendritic cells, like I said, we've published in more advanced tumors and in proof of concept in the laboratory, just using cells, feeding them antigens and looking at the cell surface, how well it's being presented on the surface when we change the sugars. We have a lot of cell-based work, and those are published works in journals like Neoplasia or Journal Immunology or other places like that, where we're studying just the basic biology of how changing these sugars on these immune cells either changes the way they behave with other effector cells like T cells, or changes the way they traffic through those blood vessels. So we're also fascinated with locking certain cells into tumors and keeping their traffic, giving them enough time to expose themselves to T cells. A lot of kinetics. Those are probably the basic steps in biology that we published more on in the last couple of years. And those would be, breakthrough in terms of basic biology. But to jump to the clinic is a couple of steps still. And so we have to look at these ways to perturb that system and make those immune cells work better with cutting edge technologies that help change cell behavior, either through these antisense molecules. So we're testing some of those out. We test them directly on dendritic cells, which are antigen presenting cells, to help them work better and present the antigen better, or in other formats. So those are happening now and pre-published, but going into new works. So does that make sense?
Yeah, it does. Thank you.
Sure.
Okay, so moving on to our project.
Yeah, tell me.
Would you like a small summary on it before I ask the questions, or are you good?
Please, yeah, I need a summary.
Okay, yeah. So essentially what we wanted to do was identify a non-invasive approach to detecting lung cancer, which is why we learned about volatile organic compounds, which are VOCs, and we wanted to test these out on possible diagnostic tools for lung cancer. And because of this, we were trying to use synthetic biology in our project to find a plasmid construct with a promoter that is activated by VOC specific to lung cancer. And obviously right now, due to the ongoing pandemic, we haven't been able to go into a lab, but we're trying to test this method out with online software that will allow us to model our idea.
Interesting. So this VOC, the only VOC, just as an understanding thing I have right now is that I've come across was exhaled, molecules that can be assayed by mass spec type methods and that correlate with lung cancer in very small quantities. as a biomarker. So I'm only familiar with biomarker. Now, to talk about a VOC, excuse me, that turns on or off or does something with a plasmid construct, is that kind of what you're talking about, developing?
Yeah.
That, let me think about this. And then the plasmid construct, is it a reporter? Is it like amplifying a signal, or is it, are you trying to use this to modify the biology in some way?
We're trying to use it as a reporter gene. Hannah, if you could correct me if I'm wrong.
Yeah, so we were intending to use this sort of construct as a diagnostic tool. So
Yeah.
like you referenced, VOCs are biomarkers for lung cancer and you can sense them when exhaled. So we are intending to create some sort of system with a plasmid that is not a plasmid, sorry, a promoter that is specific to a certain VOC, and conjugating it with GFP to sort of show.
Yeah. Very cool. Yeah, that makes sense. The question is, I guess the question is, you have to have a little bit of a pre-knowledge from early biomarker studies with VOCs to know which one to choose, like one that has, and it's always the goal in diagnostic studies to get something that has high sensitivity and high specificity. So those are the kind of things you would dream for. And if you know the VOC that's like, or a candidate VOC or a group of candidates that could turn on the, if turn on's the right word, the promoter that you're interested in, you need that pre-knowledge, which I don't have for you. I don't know which VOC is the best one on the world for lung cancer. But the point is that by searching that and looking at published data and seeing some winners, then you guys may be ahead of me on, or maybe ahead on this already, and you already know what a good candidate is, or a good family that turns on a promoter, as like a transcription factor or something, like would work the same way to just activate expression. And then you've got your GFP downstream from that. Is that kind of, am I understanding that right?
Yeah. And you did reference some of the work we have already done. So thankfully, lung cancer is one of the cancers that's like a lot of research has been done into biomarkers and VOCs. So we have looked into that, and we're planning on doing an assay of sorts. So we're choosing a couple of different promoters for different VOCs in order to ensure greater specificity, hopefully.
Great. Yeah. I mean, rest his soul. I knew a guy named Pierre Masson, who is passed away this last year, just within the last 4 or 5 months. Fairly young guy, kind of sad. We lost him in the lung cancer community, lung cancer research. He's a pulmonologist. He was at Vanderbilt and University, and he was a master at VOCs. I mean, he was totally into the stuff. Probably published quite an arsenal in it. It's not my area, my specific area of expertise, but it is I do work in a sphere. I didn't share this with you guys earlier. Clinically, I am involved in running a lung cancer screening program with low dose CT scans in our center. So I'm very familiar with lung cancer screening and early detection. And so in that program, you can imagine we have a number of nodules that we detect that are small with the low dose CT scans. So to put your idea clinically, if you develop something out of it, where it can help greatly is where you have what are called indeterminate nodules. So we detect nodules that look very scary. We can go right to tumor board, right to surgery, take care of the case. We don't even need a biopsy. Then we have nodules that are very small, and all you need is a repeat image in 6 months, because you're not going to deal with something so small. The chances that it's cancer are less than 1%. And then we get these intermediate nodules that are, 8 millimeters in size. They might be round. They might not be spiculated like cancers look early. We might get a PET scan, which often helps us see if it's got metabolic activity. This is how we deal with early lesions that are nodules clinically and help us know relative risk and whether to go to surgery and what to do with it. And where volatiles may be very helpful is in that same patient, if they exhale and we can use the volatile to enrich our screens and learn which ones are much higher risk, and we don't want to wait on a repeat image on and get active against it, and which ones we can wait on and say, this doesn't have the volatile or the volatile family that we want. And we can wait and sort of classify that group. They're often called classifiers because there's all kinds of things that do this, either swabs from your epithelium in your mouth, all the way to blood tests. They're all classifiers that help give a high negative predictive value, so high sensitivity, meaning that a negative test means you can wait longer on that screen. And that's how a lot of these technologies are being used in lung cancer detection, is that they may not be the 100% specificity and sensitivity tool. That would be amazing, right? You never get that. You're one volatile that is totally sensitive. Even the smallest lung cancer, it detects it, and it's 100% specific. That'd be the $64,000 molecule, fine. But that doesn't exist. You guys might be the ones to discover it. But if the bottom line is that if that existed, we wouldn't have a problem. We wouldn't need all these classifiers. But because they're only so sensitive and so specific, whether they're blood tests, whether they're buccal swabs of smokers' mouths, which change and put people and can help us understand whether an array that's expressed of 9 genes predicts yes or no on the risk of lung cancer when you have a nodule. That's how we apply it. Nodule's present. We don't know what to do. Are we going to get aggressive on it, or are we going to wait 6 months and re-image it? And that's a scary thing, for a patient and for a clinician. And to have some way of classifying it, saying, don't worry, this fits the signature of low risk, you can wait 6 months, versus that's called a classifier. That's how most of these are used. Versus, oh, it's a high risk signature. The volatile looks like it's telling us that there's a high risk of lung cancer here. It's a positive. Maybe we need to be more active. And once you identify a molecule like that, then what are done is a lot of validation studies to just do it in a large number of patients and look retrospectively, did the presence of the classifier correlate with what happened to the nodule when they went the next step? So your volatile could behave just like that. But I wanted to spend a minute explaining that because I can see a very nice use of volatiles, just like a lot of other things, in lung cancer screening nested in the screening data. Outside of that, you can do it without lung cancer. You can do it without low dose CT scans. If it's like the best tool in the world, you don't need a low dose scan. You can just get a volatile in a smoker and know in someone who's smoked a certain amount and say they meet a threshold for risk. I don't need a low dose CT scan. I don't need anything. I just get this volatile. It's so sensitive and specific that it's a win. I don't need and then it's a positive, you get a scan, but there it is. There's the lung cancer. That's the other way of doing it. Does that make sense the way I explained that?
Yeah, for sure. We had a couple more questions just based on your own experience. So to start off, specific to screening itself, do you think, for this kind of diagnostic test, do you think that there's going to be a huge problem or any risks attributed just to the diagnostics test specifically that you see?
I love the volatile. That's great. I mean, that's why Pierre's work and others' work, I've just really hailed because it's just an exhale. There's no blood draw. There is no, in the early days to get classifiers, we actually had bronchoscopy. You had to go with a bronchoscope, do a lavage of the lung, go measure the bronchoalveolar lavage, spin the cells down, do the array, and see if the array correlated with high or low risk on someone that has a nodule. Okay, so forget bronchoscopy. That's a lot of work and putting a patient through a lot for that one classifier. It's moved that technology. Now they can swab the inside of the mouth. Pretty low risk stuff. Blood test is pretty low risk. Exhale, there's zero risk. The only thing you have is patient identifying data, right? That's all. Whenever you deal with names and social security numbers or any of that kind of thing, you've got a risk for patient data. You always have to be careful with that, but that's what everybody deals with when they do studies. So you're at the lowest risk, level in terms of, as long as the collection chamber doesn't impose risk, and as long as it's not, I think the highest risk is where it's misleading something, where it starts, the data derails you and you feel that something is not cancer and it is. So, it's giving you its sensitivity and its specificity is poor, so it gives you false positives or false negatives. That's the real risk in that kind of data, but that's where you have to just validate it in prospectively in large patient sets. When you discover something that you think correlates with lung cancer and it's a volatile, you take 100 patients that have a risk for lung cancer, like a nodule on a low dose scan, and look, do the volatile test and watch what happens with time. But you don't make clinical decisions with it. You just watch. And if you see that, my gosh, that correlates perfectly. Everybody who has the volatile had cancer, everybody who doesn't have cancer, then you're safe to go the next step where an IRB or a review board or anybody that allows you to adjudicate the next step to say, we can now become clinical with this and start using it to make clinical decisions after the validation studies, then it's kosher to go there. But those are my risks. Is my biggest risk with a volatile is jumping too early to misinform and erroneously classifying patients, if that makes sense.
Yeah, it does. Thank you. So I have two more questions. So again, based on your experiences with these like various patients that have had like lung cancer or being tested for it, do you think that they would be willing to take or trust this type of test, especially considering like the how delicate like the volatile organic compounds are?
Yeah, I don't have any, like in terms of trusting having it done. Is that what you're saying? Like,
Yeah.
no, I don't have any real concerns there. That once you just have to be extremely careful with, first, anything you do like this has to go through an adjudication that you're allowed to do the study. Remember, you're going to get a patient to sign a consent to collect breath, and there's patient data involved. So you're handling a name, and any clinical data that goes with that is just in terms of running studies, what we have to get clearance from is an IRB or an institutional review board. And so when you're going to have real patients going through this, and an IRB will read about your proposal, you'll propose it to them, they'll say, yeah, looks safe, patient trust, et cetera. I don't have a problem with the patient's trust in this one. It's not like the dangers of like, suffocating during a bronchoscopy to get the sample. You're just blowing onto something. So not a big deal. But in terms of patient information and how you use it and getting the patient to consent that your data is going to be used and potentially at risk of being disseminated or falling in the public, or someone loses the form that's locked in a desk that has the patient numbers and the data, that's probably the bigger one. And then the intermediate one is that they have to know that how is their data being used to help themselves or other patients. And many of these things don't help the individual. There are many studies. They're just great that they're helping the public to get data, and they're participating to give biological research growth so that therapies can be developed. And so that could be a typical thing, is that your data will be part of a validation study. And that could be a post IRB kind of adjudication. Yes, you're good to go. That's how the patient's going to be used after they sign a consent. And so does that make sense? It's a consent process.
Yeah, it does. Thank you. So one last question. I feel like you kind of already answered it, but do you foresee any other ethical concerns with our diagnostic tools other than like releasing the data to the public?
Not really. It's the ethical concerns come more in handling data. Releasing is also ethically can be dangerous if it's not released with the right biostatistics applied, with the right accuracy. The gathering's the more dangerous part. I think it's more that their information is at risk. Their name with their volatile or their, whatever it is something that's being used to get valuable data that they've consented to. And that whole consenting process, as long as you're going through that the right way, that's all cool. I just think at the end, in terms of presenting the data, where you have to be ethical too, is in, digesting it. There's no fudging, there's no cherry picking. It's fair. It's done with a biostatistician often. That's where we go to a lot of times for help when we have large amounts of data, where you give the data, and it's all numbers. Nobody's name is on there, 1 through number 80, whatever. You turn it in, and number 56, number 85, whatever there. It's all de-identified, we say. And then when you run the de-identified data and analyze it in the analysis, you have to be ethical about working with proper statistics, reporting exactly what it is. And you can't hide data. You can't, it is what it is. And you do the correlations, and you come out with real data that you can report in a paper. And then, that's where you go the next step, which is the reporting of it is ethically important. Whether it's negative or positive data, to be able to report it is very helpful to the public, because you've learned something either way. So.
Okay, thank you so much. If Hannah or Flora, do you guys have any more questions?
This isn't really an official question so much as it is like personal bias question. So I saw in your bio thing that Flora sent over to me and Shefali that you were an MD. So I was wondering a little bit about your educational background, and how I haven't seen a lot of medical doctors break into research. So I was wondering what steps you took between there.
Yeah, so it's pretty interesting if you go into academic circles in the university, and you're in health sciences. So that's a large group that has Department of Medicine, Department of Pediatrics, all these departments that do anything that touches medical science in it. You'll actually find a lot of PhDs. You will find a lot of MDs too. MDs that do biomedical research are, in the larger numbers, are just MD, interestingly. There was a period, and there still is a period, it's still going on, where there's people doing MD PhDs. So we call these clinician scientists or physician scientists. And traditionally, there have been many more physician scientists that are MD alone than MD PhD. I think MD PhD was a great invention, because it just helps somebody who's going to spend many years in a lab learning the tools and doing what it takes to learn the biology, if they're a cell biologist or a molecular biologist or a biochemist. It gives them a thesis, it gives them classes and all these kind of things. The MDs that do it, they don't train like you're thinking, where you're probably thinking, how's this Dr. Mark going through medical school and just jumping right in the lab and taking cells and doing, genetics in mice and in cells and right away? Well, no, it's not a finger snap. You don't just kind of do your MD, see patients, and then just open a lab and start doing it. So I trained by a mentor, just like any PhD would, and did a postdoctoral period of, and I had to get grants in it. So we write grants, they're career development grants, and these career development grants fund my protected time during my development after my MD, actually after my residency. So I went into my specialty, then during my fellowship, which is like the period where you train as an MD, as a subspecialist, train how to be a pulmonary and critical care expert, which is what I am. That's what I'm boarded in. That's my type of MD medicine. You've heard of cardiologists, so I'm a pulmonologist, but nephrologists, they deal with the kidney. Rheumatologists, they deal with joints and connective tissue disease. And so I'm a pulmonary and critical care doctor. So I have that type of medicine. When I was a fellow, which is 3 years of specialty training to do that, they require half your time to be research. And you can do that research and leave and go into practice. But a good chunk of us, maybe a third of us, keep on in the university setting and use that training to go further. And so my training went beyond that, my science training. So I was literally in a basic science lab. Many do clinical research, many do clinical volatiles that say, that's not always, you're just taking data that comes out of a lab and using it. But some of us do basic research. So we're working with literally cells in a lab, putting molecules on, watching how the cells roll, or interact, genetically affecting them, doing biochemistry. That's the way I grew up, in someone's lab who was my mentor for many years. And so outside of fellowship, beyond it, I went in as an attending physician. So now I'm done and I'm overseeing residents, fellows in the academic system. And while I was doing that, in good 70% of my hours, I was in a lab, still training as a postdoc for 4 or 5 more years, doing just the same kind of science every day as a PhD would do in training. And that's just what many MDs have done who have become physician scientists. And so it's the majority of people that have MD next to their name that just have the MD and do science versus, I mean, I said that wrong. There's many more MDs that don't do science. They're purely clinicians. But of the MDs that do, that pool is yay big, whereas the MD PhDs, it's quite a bit smaller. And that, if that makes sense.
Yeah, it's really interesting. And if there's 7 more minutes,
Yeah.
I don't know how much in a rush you are. So in your experience and maybe other people you know who transitioned from being MDs to like working in the lab, so did you initially think that you were going to go into research, or did it just like end up being a passion that you developed over med school and over your residencies?
Yeah, mostly over time it developed. I kind of thought it would probably be, I guess when I was a resident, that's after medical school, I was kind of open to kind of halfway. I sort of like curious in terms of the type of science I would want to keep doing versus just being a pure clinician. And the kind of specialties, the kind of world that interested me was kind of between lung injury centered and cancer. And I've kind of continued that on. So there is a part of my lab that we study viral lung injury. So we do COVID research. We do flu research. You know, before COVID hit, we were doing viral lung injury, and some of that same genetics and cell biology we apply to that world. And part of the whole. So in my early training, I didn't know I would become those sort of like interests in lung injury and in cancer biology. As I went into the specialty of pulmonary and pulmonary critical care, I sort of took the half of me that said, you know what, I want to keep doing this as a science and get it to the patients one day, because, you know, there's a lot of frustration in terms of the inability to treat a lot of things, and look how much death we deal with and suffering with insufficient medicine that we could make much better. So that part stuck with me. And so that's where I kept going. And it just turns out that those curiosities are kind of what I'm still doing right now in terms of studying in the lab. Those are heavily parts of what I study. So
Yeah. And do you think your background as like a trained physician have like affected your perspective towards science as a whole and like what you bring to the lab?
yeah, I mean, I think that the goal, that it's a kind of it's always made it sort of I see it very simply that the goal of my research is to, whether it happens in my lifetime or beyond, bring it to improving patient care. That's all it is, but at a very basic science level. So if we make discoveries that help the next steps in a number of technologies that help patients with cancer, help patients with viral lung injury, great. That's the goal. So it's pretty straightforward. I don't really study things for the sake of curiosity only. It's more directed to improving health.
Yeah, it's very admirable. So Flora and Shefali, do you guys have any more questions?
Flora, you were kind of quiet here. Do you have any any interesting questions or anything I can?
I was mainly just like a connection between these two aspects. I don't really have anything. Maybe like based on what you've heard of our project so far, do you have any other like feedback, or do you have any ideas of maybe what else we should be looking into?
Yeah, I was wondering that. So good question. Yeah, I do. I just was wondering kind of where, you know, I was still kind of feeling out where you picked a couple of candidate volatiles and why those, or, you know, or maybe ask better, is it more the tool that's driving you, that you have a promoter and a potential volatile, or is it more the volatile that's driving you, the volatile candidate, and you'll make the promoter work no matter what it takes? So I don't know if that makes sense. Like sometimes the platform is there and the question is modified a little bit so that it fits the platform. So you might have a great promoter and a great reporter system, and you kind of are willing to take volatiles that turn that one on, even though they're not the greatest in terms of what are candidates, but it proves that the system works, that you have, and there's nothing wrong with that. That's actually discovery technology. You can do that, and you have a technique then that you've proven, even if the pre-test probability of that volatile isn't the greatest for lung cancer, but it's okay. Versus, it's like the horse, the cart and the horse driving the, you know what I'm saying? Like, versus, you know what? I'm going to pick the best volatile and we're just going to make the system downstream work for it. I don't care what it takes. We'll make a promoter work. We'll make a GFP. We'll just, and both are right answers. I just want to get a feel of what you feel you're at with that. Does that make sense? I don't know if, or anybody, I'm not asking you, Flora, just anybody in the group, if my question makes sense, or maybe you're not even there yet. You're still in like the design step of, we need to pick up a volatile for lung cancer.
Hannah can correct me if I'm wrong, but I think right now what we're doing is each person in our team is looking at like a volatile and promoter pair, if I'm correct. And I don't think we've decided on anything specific yet. Hannah, you can add more into that.
That's great. Yeah, add more if you can tell me more there.
Yeah, so we aren't working in the wet lab this year. We're mostly just doing design and the like. So sorry, it's gonna take me a second to pull this up. Okay, we've narrowed it down to, I think, 4 promoters that have been shown to bind to these volatiles. And I think among them are like astine, toluene, styrene, and 1 more. So we're using these 4 that have been proven to work before and sort of synthesizing them into 1 plasmid.
Okay. Great. Yeah, I think the goal is awesome because you would be able to amplify the signal and you won't need a mass spec or, you know, one of these fancy systems, right? Am I reading that right? I mean, you just have a way of fluorescently reporting.
Yeah, that's the goal. And I guess a future direction would be looking at the degree to which this fluorescence happens.
Okay.
Based on like concentration of the volatiles we're looking at, like concentration in the breath, and
Yeah.
how sustained this fluorescent signal is. So that's a future step if we get into the lab.
Yeah, like a quantification. I get it. That makes sense. Like presence or absence is one thing, but having enough, that's interesting to quantify. That's the transduction is the interesting part here, how you get the volatile to turn on the promoter and what that means in terms of a quantity thing, you know, is it like 1-to-1 or, you know, like what does that translate into? Because amplifying a GFP signal could suddenly be non-reflective of the amount that actually was there to turn it on.
Yeah.
So those are good questions. And I'm sure there is some molecular biology type, you know, plasmid gurus that probably would help with that better than I can help you.
It's definitely something to look into, and we haven't thought about before for sure. So
yeah, great.
We should be pointing it out.
Okay.
It is 4:01 p.m. Does anyone have last notes? All right. Well then, thank you so much for coming and talking with us.
Hope I've been of help. Where does this go? Do you guys like do a presentation on this, or how does this?
Yeah, so the way iGEM is structured, it's a synthetic biology research competition.
Yeah.
So we've talked about our project already, and it's going to culminate in sort of a poster presentation around, I want to say October or November, where we're just going to present what we've been doing and in front of a panel of judges, and they can just give us feedback on our project. It's, for us as high schoolers, it's just a way to get involved in the scientific process. And I think it's super enriching for me at least, so.
Yeah, that's great. Well, I probably wasn't very helpful for your project, but if it's any guidance that there's pathways that you can take your project and get it to patients one day and give you some paradigm of going through different medical or PhD pathways, happy to be an example. I don't know what to say.
Yeah. I loved hearing about your research though, in case you couldn't tell from the stupid questions.
Yeah, of course. Yeah.
All right.
Totally. I'm glad you like it. Okay. Great.
Thank you so much for coming then.
Thanks. Take care. Good luck. All right.
Thank you.
Oh, Shefali left too?